Thank you to Alex's for inviting me here to speak today and for all the support. I'm really excited to share some of our work today. So, you know, as Jay mentioned, the topic of my talk is really how we unleash the immune system against pediatric cancers. So I'll divide into three basic parts. First, just a short overview of immunotherapy for pediatric cancers. And then our initial efforts to bring CAR T cells forward for pediatric solid tumors, and then some new work into unleashing the innate immune system against neuroblastoma and osteosarcoma. So when we think of immunotherapy, we really divide it up into kind of two different worlds. And there's this one on the top, which is immune response amplifiers, and then the second one, which is synthetic immunotherapies. So first, I'll just talk for a little while about immune response amplifiers. And this really, when you ask an adult oncologist about immunotherapy, this is where they go, and really because of checkpoint inhibitors. So these are agents that are able to unleash the body's natural, the immune system's natural response to tumors by unleashing T cells. And, you know, we were very hopeful initially, we were initially hopeful that these agents would also have some activity in pediatrics. But, you know, they basically unleash T cells, and T cells need something to respond against. And T cells are basically programmed in order to respond against mutated or foreign peptides presented by tumor cells or infected cells. And so basically you need a lot of mutations for T cells to start responding. This is work from Adam Schline's group in Toronto, where they looked at the number of mutations in pediatric cancers. And what you can see is that it's really only 1% of pediatric tumors that have very high mutational rates. And these are the types of rates that you see in responders to PD-1 therapy or checkpoint inhibitors in adult malignancies such as melanoma and lung cancer. And most pediatric cancers have much lower rates of mutation. And, you know, kind of in agreement with this, we also looked at PD-L1, which is the ligand for PD-1. We looked at expression of PD-L1 on about 400 pediatric tumors. What you can see here is in those common pediatric sarcomas and here medulloblastoma and brain tumor, you see very low expression of PD-L1, which is generally thought to be, not always, but generally thought to be an indicator of your potential to respond to anti-PD-1 therapy. And so, not surprisingly, as the phase 1 trials of PD-1 were brought forward, both through COG and other cooperative groups, this is one that was led by Kara Davis and Crystal Makel through COG of nivolumab. We really, unfortunately, did not see any responses in these common pediatric sarcomas or neuroblastoma to single agent checkpoint. So for most pediatric tumors, because they're not highly mutated, and they're like Ewing sarcoma, rhabdomyosarcoma, driven by single mutation, single gene fusions, or as in other tumors, some epigenetic changes, we really are not seeing robust responses to single agent checkpoint. Now, this doesn't mean there's no role for checkpoint in pediatric cancers, and now interesting combinations are coming forward. And then also there is a small subset of patients who have a deficiency in their DNA repair mechanism, something called biallelic mismatch repair deficiency. And the group in Toronto, led by Ori Tabori, really nicely has shown that those patients who have hypermutant cancers are very likely to respond to checkpoint inhibitors. But as we think trying to extend out to the large population of patients that have these solid tumors, we've started to think differently about how to initiate immune responses. And so rather than immune response amplifiers, what we've tried to focus on is these synthetic immunotherapies that will initiate new immune responses. And these really, at a most basic level, can include things like tumor-specific antibodies, bispecific antibodies, but our group really focuses on CAR T cells. So just as a reminder for everyone, CAR T cells basically allow you to endow a T cell with the specificity of an antibody, but the cytolytic capacity of a T cell receptor in a MHC unrestricted or genetically unrestricted manner. And these agents have really revolutionized the care of pediatric leukemia. And this work comes from Shannon Maud and through the trial by Novartis, the Eliana trial, that led to the FDA approval of CD19 CAR in pediatric ALL. And there were just, amazing complete response rates to a single infusion of these therapeutics. And this is the first patient, Emily Whitehead, who was ever treated with our former president, Barack Obama. And, this work really came out of basic lab work, understanding how to make these therapies that happen all across the country at multiple institutions, including University of Pennsylvania, the NIH, Sloan Kettering, Seattle, City of Hope, et cetera. And really just came to this moment where people realized, oh my God, this can work in pediatric cancers, or this can work in cancers in general. And we just saw response rates that were unheard of for phase one clinical trials that led to their eventual multi-center trials and FDA approval. Now, I came to my fellowship around the time that Emily Whitehead was initially being treated at CHOP, and I was at NIH with Crystal Makel, also running a trial of CD19 CAR. She was running a trial of CD19 CAR in pediatric ALL, along with Trey Lee. And I had my first year on the wards in the NIH and never thought I would go into the lab, but saw what was going on and just the amazing power of these therapeutics. And Crystal fortunately recruited me to come to her lab. And since then things have really taken off in this world, and it's been amazing to watch how these therapeutics have developed. Now, when I joined the lab, I said, okay, I joined Crystal really to bring these therapeutics forward, CAR therapeutics forward for solid tumors. And we hoped that it would be pretty simple, that we would just switch out the antibody on the CAR, and we would see the same type of responses that we had seen in leukemia and lymphoma in solid tumors. But first, we needed a good target. So when we went looking for a target, we wanted to look for something that was present on tumor cells and absent on normal tissue. And that's because a CAR T cell will attack any cell that expresses that antigen. Fortunately, in the case of CD19 CAR, the antigen is shared only with a non-essential tissue, normal B cells. And for solid tumors, we're not as lucky because most of the tumor-specific antigens are also shared with the tissue of origin. So we wanted to look for something that was largely absent on normal tissue, and that search led us to ALK, which is a well-described oncogene that's overexpressed on neuroblastoma and some other pediatric cancers. So we looked on our neuroblastoma cell lines and said, great, some of them express ALK at moderate levels, let's keep going. And we basically just took the Kymriah backbone and flipped in a different antibody, this one recognizing ALK that came from collaborators in France. And, we were hopeful then, okay, we were just going to put it in the mice and it was going to work. And unfortunately, it wasn't so simple. These are two models of neuroblastoma. And what you can see is that in red, the CAR T cells do mediate an effect against the tumor cells, but ultimately they're not that effective. They're nowhere close to what we had seen with CD19 CAR in mouse models or in children. And, ultimately the tumors all grow out and these are non-curative. So we really wanted to understand what was limiting the efficacy of our CAR T cells. And, the first thing people kind of talk about is if it's a solid tumor, there's a problem with the tumor microenvironment. There's suppressive cells in the microenvironment that keep your CAR from working. And we were more interested in thinking about, okay, how can we improve actually our receptor? And what factors actually influence the behavior of our anti-ALK CAR, our ALK receptor? So what we did to kind of take out the question of the tumor microenvironment was we took this NOM6 cell line, which is a leukemia cell line, and then we used a lentivirus to overexpress ALK on its surface. We then FACS sorted and single cell cloned to obtain clones with different amounts of ALK on the surface. And here you can see three of those clones, in a high, a medium, and a low. And the low is specifically selected because it approximated the amount of ALK that was on many of our neuroblastoma cell lines. And then when we went to look at the killing of these tumor cells in response to our CAR T cells, you can see that it's really proportional to the amount of ALK on the surface. So when there was ample amounts of ALK on the surface, as seen here in blue, you had robust killing. This was in line with what you would see from this assay from the CD19 CAR T cell. But as you decrease the amount of ALK on the surface, you had much less killing by these CARs. And when we looked at cytokine production, the results were even more interesting. So here you see the amount of IL-2 produced by the CAR T cells in response to tumors of varying amounts of ALK density. So as you go across the x-axis, you see rising amounts of ALK on the surface. And what you can see here is that there's really a threshold antigen density you need in order to start generating ample amounts, substantial amounts of IL-2. And in all of our models, IL-2 has usually predicted the efficacy of these agents. The production of IL-2 has predicted the efficacy of these agents in mouse models. So we found a threshold of about 25,000 molecules of ALK needed on the surface of a tumor cell to start producing substantial amounts of IL-2. And unfortunately, most of our neuroblastoma cell lines expressed ALK below that level. So we then took it in vivo, and we took leukemia that either expressed a high amount of ALK or a low amount of ALK on its surface. And you can see the ALK CAR mediated really substantial effects against the high ALK expressing leukemia and nearly no effect against the low ALK expressing leukemia. And against the high ALK expressing leukemia, the ALK CAR started to look a lot like the CD19 CAR. So, this led us to say, okay, clearly we need a target that's expressed higher. We either need to redesign our CARs, or we need a target that's expressed at higher levels than ALK. And so we kind of reoriented our search. Now, one of the first criticisms we heard about this work was, well, that's just, you made a crappy CAR and other CARs don't do that. And we heard that CD19 CAR doesn't do that. So in another manuscript, which I really won't get into today too much, except to say we generated clones of NOM6 that express different amounts of CD19 on their surface. First by using CRISPR to knock it out, and then reintroducing it with the lentivirus again. And then we tested the commercially available CD19 CAR T cells against these different clones. And both in vitro, which is shown here, and in vivo, there's a clear loss of efficacy when you drop, this is a killing shown along the y-axis. And as you go against these NOM6 clones that express about 1,000 molecules of CD19, you see that Kymriah, the CD19 41BB CAR that's FDA approved for B-cell ALL, no longer is able to control the tumor. So we really took this as, all of this data together led us really to say that CAR T cells have a deficiency in being able to go after low antigen density tumors. This stands in contrast to native T cell receptors, which are very, very potent and sensitive and can recognize as low as 1 to 10 peptides in MHC. Instead, we started seeing CARs needing 1,000 to 3,000 molecules of CD19 on the surface for efficacy. So this really reoriented our search for new targets for CAR T cells in solid tumors. And I'll show you why. So the first type of target, which is really the ideal target, is one that's absent on normal tissue or vital normal tissue and high in tumor. And CD19, CD22, et cetera, for B cells really fits the bill. And that's because humans can live without their B cells if they get replacement of immunoglobulin that's made by those B cells. And no vital tissues express those targets like CD19, and it's very, very high on BALL. So this is really one reason CD19 CAR has worked so well, and CD19 is such a good target. Then you have targets like ALK. So this is where we started when we started looking for things in solid tumor, where we wanted things that were absent on normal tissue, but we really only found them to be moderately expressed on tumor tissue. And so those clearly would be safe, but were they promising? And then you had this other group of antigens that have low expression in some vital normal tissues, but are extremely high, screaming high in tumor. And we had previously thought, okay, maybe those were too dangerous. But given what we were seeing about CAR T cells, that they're not able to recognize, at least our current CAR constructs are not able to recognize low antigen density, we realized that there may be a therapeutic window for these type of targets, the targets that are extremely high on cancer, but still expressed at low levels on normal tissue. And we started looking for targets like that. So that was right about the time that we came to Stanford. And, you know, we had the fortunate experience of getting to know Michelle Manji, who's one of the world's experts in disease diffuse intrinsic pontine glioma, which is a high-grade glioma, a high-grade brain tumor that, that occurs in the brainstem of children. So it's, you know, it can't be surgically removed. It's extremely aggressive. The median life expectancy is less than 10 months, and less than 1% of patients survive for 5 years. So this is really a incurable pediatric brain tumor, and there are no real known therapies other than radiation, which is really just palliative. So Michelle Manji's group had started looking at immunotherapeutic targets on post-mortem biopsies that she has made it her life's work to obtain from these patients and as a gift from their families, so we can really understand this disease. And interestingly, what they found was that this target, GD2, which is a known target in a very well-known target in pediatric cancer, especially in neuroblastoma, but that it was expressed on DIPG. And so on arrival to Stanford, they asked us, oh, do you want to you know, see if the GD2 CAR would work in DIPG? We said, okay, sure, give us some of your patient-derived cell lines, and we'd love to see how much GD2 is actually on their surface. And what we saw really blew us away. And I say that because you can see the GD2 expression for these common diseases, neuroblastoma, glioblastoma, and osteosarcoma, where GD2 is known to be expressed. And DIPG always expressed levels that was at least a log higher than many other of the pediatric tumors. And this was really interesting to us that it was so much higher, and this is on a log scale, it's so much higher on DIPG than even in most neuroblastomas, where GD2 is really a validated target. So, you know, we were excited then to start testing CAR T cells in these models. Now, why do we think GD2 is so high on DIPG? So DIPG is basically driven by this mutation, a mutation in histone 3 that results in hypomethylation and transcriptional dysregulation. And this happens throughout the genome, but specifically the genes that are involved in making GD2. So GD2 is not a protein, so you don't have a 1-to-1 ratio, or you don't have a 1-to-1 question that you just need to increase one mRNA and you'll get more protein. It's actually made by a complex step of with many ganglioside synthase enzymes, and those synthase enzymes that are involved in the production of GD2 are actually expressed very high in histone 3 mutated tumors, and in histone 3 wild type tumors hardly expressed at all. So we realized that this is really a target that is driven by the disease itself. And then when we went and we took GD2 CAR T cells and tested them compared to CD19 control CAR T cells, we saw really, these are intravenously administered GD2 CAR T cells. We really saw profound activity of these CAR T cells compared to unrelated control CD19 CAR T cells. And, you know, around day 21, Chris Mount, who was the graduate student that authored this work with me, and my partner in all this, came to me and he goes, oh my God, all the tumors are cured. And we said, yeah, we know that's how CARs work. But for them, it was, you know, we were obviously excited about the data, but for them, they had never seen this level of tumor clearance from any therapeutics tested before. So we really saw really enhanced survival in mice that were treated with the GD2 CAR T cells. And just to put this into context and compare to a group treated with panobinostat, which is a emerging therapeutic in the clinic right now, you know, that basically extended the survival of mice by 12 days, whereas here, all of the mice had long-term tumor control. So we were really excited about this data. And then we wanted just to take a closer look at how these CAR T cells were entering the CNS and what tumor clearance looked like. So we generated these CARs, either GD2 or CD19 control CAR, that express 4-1BB zeta, which are the signaling domains, and then this fluorescent protein at the end, M-cherry, which allowed us to track it by confocal imaging. And what you can see on day 7 after we treat with the GD2 CAR, you can see in magenta, the CAR T cells have all collected outside of, in the parameningeal region, ready to, or starting to enter the brain to clear this GFP positive tumor. By day 14, they've permeated the entire pons and cleared all signs of GFP positive tumor. And by day 21, they start contracting. And this stands in contrast to mice treated with the CD19 CAR, where there's really no infiltration of CD19 CAR T cells into the CNS and perfuse tumor throughout the pons. So we've now, fortunately, with the support of PICE and CureSearch and some other donors also, we've been able to translate into this clinic. And I'm happy to say we're now treating patients on this trial that I'm co-leading with Michelle Manji here at Stanford. And it's very early days, but really, we're really excited to see something that was taken quite quickly within 3 years from the laboratory into the clinic. So, GD2 is a great target in DIPG, but it's not as homogeneously expressed in other pediatric tumors. So we've also been looking for another target. And of course, we look at tumor-specific targets, but when you think about pediatric diseases, which all of them are rare diseases, relapsed Ewing sarcoma, we're talking about less than 100 patients a year. What we really want is a target that we can go after for multiple cancer types. And what I think we found it in B7H3, and I shouldn't actually say we found it, because this target was really defined as a pediatric cancer target and a cancer target in general, really by the group at Sloan Kettering, who has been targeting it in CNS malignancies with a monoclonal antibody. Antibody radioconjugate called 8H9. And so this was a really known target, but we were happy to see that when we looked at about 400 pediatric tumors, over 85% of them expressed B7H3, and mostly at really high and homogeneous levels like you see here. We see it in solid tumors and of the periphery and CNS tumors. So this is also a target in adult malignancies. And this is work that came out of Macrogenics, a company that has a monoclonal antibody targeting B7H3. And they made this antibody, MGA271, and selected it from hundreds of antibodies because it's really, they found specific for when B7H3 is overexpressed on tumor cells. So here you see a panel of it from their manuscript of a panel of adult malignancies that stain really well with this antibody. And then on normal tissue, there really was almost completely absent expression of B7H3. So here we just did some plug and play. We actually tested 10 different antibodies, but this one from Macrogenics was actually the most effective. But we did a plug and play kind of into the Kymriah backbone and made this CAR T cell. And unlike the ALK experience, this one really worked well. So this is an osteosarcoma model where we treat, once the mice have pretty substantial tumors, around 21 days after we implant the tumor. And you can see that the B7H3 CAR is able to completely regress this tumor, whereas the CD19 CAR, which is the unrelated control, has no effect and the tumor keeps growing out, and this results in long-term survival of the mice. We saw the same in Ewing sarcoma. We've seen the same in other malignancies as well. And then we started looking at brain tumors. So here we inject the brain tumor directly into the posterior fossa of the mouse. This is a medulloblastoma. And then a few days later treat with CAR T cells. And what you can see is that the B7H3 CAR, again, has this profound effect in most mice to clear the tumor. And results in really prolonged survival for those mice that are cured. Whereas the mice that do not receive CAR T cells die within, or receive unrelated CAR T cells, die within 20 days. So this is work that we published in 2019, and then the following year, Johanna Therovath in Crystal Makel's lab did some follow-up work that looked at the ways that we can administer B7H3 CAR T cells to treat brain tumors. And she did this in models of ATRT, which is another highly malignant and highly deadly pediatric brain tumor. And she compared the method of injection of the CAR T cells to clear these xenografts. And what she did here was she compared mice that were treated with IV administered CAR T cells versus ICV, when the CAR T cells are administered directly into the CNS on the mice. And what you can see here is when they're administered directly into the CNS, into the spinal fluid of these mice, you see really fast tracking of the CAR T cells seen in green here to the bed of the tumor. Whereas 2 days after treatment, you don't see any CAR T cells yet get to the brain of the mice that received intravenous. Fortunately, also the ICV therapy, this direct administration of CAR T cells into the central nervous system, is associated with lower levels of cytokine, which hopefully would mean lower levels of toxicity in clinical trials. And then when you look at efficacy, you see that these tumors are curable with IV administered CAR T cells. Here we use 10 million. But when you go ICV, when you administer the CAR T cells directly into the spinal fluid of the mice, you actually can cure with 1/10 of the dose, 1 million. And when you use that dose peripherally, the CAR T cells have no effect. So this really seems to point that for certain brain tumors, it seems to be highly advantageous to use this regional delivery of CAR T cells. And this is being tested by multiple groups, both in children and adults in the clinic. So, to conclude the section about CAR T cells, I'll say that they really have revolutionized the care of children with relapsed leukemia, and now are being brought into first line or second line for pediatric leukemia. And we're very excited to see the data from those emerging studies. And they're really poised for major breakthroughs in solid tumors. And the target selection should really be about finding things that are expressed homogeneously at very high levels. So our GD2 CAR, we believe, is an exciting approach in DIPG. We're currently treating patients on a phase 1 trial at Stanford, and we'll soon initiate a multi-center trial in neuroblastoma and osteosarcoma, where GD2 is also known to be expressed. And I just want to say that these centers, both Texas Children's has multiple GD2 CAR programs, UNC, Rome, and there was a group in London had one recently, all have currently open trials for GD2 CAR in neuroblastoma and or osteosarcoma. So it's great to see those therapeutic options coming to light for patients. We think B7H3 is an exciting pan-cancer antigen that we can deliver either systemically or locally in the CNS to treat brain tumors. We're currently planning a phase 1 trial in brain tumors, and the group in Seattle Children's has really nicely moved forward with several exciting CAR T cell trials, including one testing a similar CAR to the one we showed here. So really excited to start seeing, for that to be an option for patients and to start seeing the results from those studies. So for the last part of my talk, I want to reorient away from the synthetic immunotherapies and back to the immune response amplifiers. So, we had written off immune response amplifiers because we said there's no native T cell response in kids for us to unleash, and checkpoint inhibitors haven't been working. But there are other cell types in the immune system. So this is a paper where the group compared the immune infiltrates between pediatric and adult tumors. And as expected, when you look at T cells between adult tumors and pediatrics, you see a huge difference, much higher T cell infiltrations in adult tumors. But when you look here, CD68 on the myeloid cells, you actually see much higher levels of macrophages and myeloid cells in pediatric tumors versus adult tumors. So we've always known that there are high levels of myeloid infiltration in many pediatric sarcomas, neuroblastoma, other solid tumors, and we've never really known what to make of them. So, another fortunate thing for coming to Stanford was learning about the CD47 story that really emerged from Irv Weissman and Robbie Majzner's labs. And CD47 is basically what they call a don't eat me signal. It's an anti-phagocytic signal that basically tells that tumor cells or normal tissue put up to tell a macrophage, don't try to remove me or don't eat me. And they generated the first monoclonal antibody that can actually block that interaction and drive tumor cell phagocytosis. And they brought that, fortunately, to clinical trials, which I'll talk about in a minute. So, you would think, since normal cells also express CD47, that when you block with an anti-CD47, macrophages are going to start going after your normal cells. But really, there's another part of the equation, that there are these eat me signals So the dominant eat-me signal in human cancers is calreticulin. And this is a protein that's usually in the endoplasmic reticulum inside the cell hidden, but it's shuttled to the surface when the cell is ready to be removed. And dying cells increase their level of surface calreticulin to signal to macrophages, eat me. And it's basically programmed cell removal. And normal tissues don't express high levels of calreticulin and these other eat me signals unless they go under stress. So the times you start seeing calreticulin high on cells, Irv Weissman's group has shown that this happens on dying cardiomyocytes after MI, et cetera. So times when cells are really ready to be removed by macrophages, they'll upregulate calreticulin. So for anti-tumor activity, when you think about a macrophage, it really lives in this balance between don't eat me and eat me signals. So on one end, the cancer cell will express CD47, which interacts with the SIRP-alpha receptor on macrophage and sends a don't eat me signal to the macrophage. Meanwhile, you have prophagocytic signals like calreticulin or the FC portion of an antibody that will interact with the FC receptor on a macrophage and drive towards phagocytosis. So, kind of the first big hit clinical trial that came out of the CD47 work was in an adult lymphoma trial where the group, this started kind of in-house in Stanford and then was eventually spun out to 47 Inc., which is now owned by Gilead. But what they found was they took adult lymphoma patients who were rituximab resistant. So rituximab is an anti-CD20 antibody that recognizes lymphoma cells. And these are patients that pretty much had all seen rituximab before. And all they did was give them rituximab with an antibody that blocks CD47. And they actually saw, around the 30% complete response rate for these highly pretreated lymphoma patients. So this was pretty exciting data when it came out. People were really impressed. These are off-the-shelf antibodies that had response rates that were similar to that being seen with CAR T cells in lymphoma. And so when I saw this data presented at ASCO, I said, okay, what's the only antibody we really use in pediatrics other than rituximab? And I personally treat neuroblastoma patients and thought about anti-GD2, which is in the clinic, FDA-approved drug known as dinutuximab. And so what we decided to do was just try the same type of combination of anti-GD2 plus anti-CD47 in mice that had neuroblastoma xenografts. And so here you can see we took mice and we treated them either with individual antibodies or the combination of both. And what you can see is that individually CD47 really has no effect. Anti-GD2 has no effect in the larger tumors and minimal effect you'll see in the smaller tumors, whereas the combination of anti-GD2 and anti-CD47 really unleashes the macrophages against these tumors and results in long-term tumor clearance. And here you can see even those mice that had minor effects from the anti-GD2 that had small tumors, the tumors eventually grow back. Whereas the combination anti-GD2 and anti-CD47 is just really synergistic and potent and results in long-term clearance of these cancers. We see the same exact results in a metastatic neuroblastoma model where the tumor is located in the liver and the bone marrow. Again, we see clearance and long-term survival only of mice that get anti-GD2 and anti-CD47. So, this was the rare project that you start with an in vivo experiment and it works really well. And then you need to go back and figure out why it works so well. So in this assay here, what we did was we took human macrophages and we exposed them to tumor cells that are stained with the dye so that you can define the tumor cell population. And here in the upper right-hand corner is the amount of phagocytosis that you see. And what you can see is when we coat the tumor cells just with anti-GD2 or anti-CD47, they have some effect to increase phagocytosis compared to the no antibody control. But when we give both antibodies, we get this kind of highly synergistic effect where we see much higher levels of phagocytosis. And we tried this with another antibody. In this case, we used an anti-B7H3 antibody. And here we didn't see any of that synergistic effect. And we also brought this into mice and used the anti-B7H3 antibody in combination with anti-CD47. And as you can see here, it doesn't look any different than anti-CD47 alone. And it's only this combination of anti-GD2 and anti-CD47 that cures the mouse. So this told us there's something specific about what GD2 is doing as opposed to using any tumor-specific antibody. So, the first thing that we looked at is something to do with eat-me signals and calreticulin. So anti-GD2 has been reported for a long time to have direct effects on cells, on tumor cells that have high levels of GD2. So here we took 2 GD2 high cell lines and incubated them with the anti-GD2 antibody, and you can see brisk cell death. This is after 2 hours in response to the antibody. And this process is poorly understood and is now a focus of my lab. But if you look at the cells that survive, so that 50% of cells that don't die, you also see that they really highly upregulate the stress signal, the eat-me signal, calreticulin, basically signaling to macrophages to please remove me. So we think this is part of the equation of why the anti-GD2, anti-CD47 combo is so effective. But there's another arm to it also. So GD2 is basically a combination of a long fatty acid that integrates into the membrane with a sugar that sticks out of the membrane. And that sugar contains sialic acids. And nature has basically endowed immune cells with several of these Siglec molecules. And they're just receptors on immune cells that bind sialic acids. And there's here, here I think 12 of them are shown, and most of them contain inhibitory signaling motifs. So meaning that if you ligate one of these Siglecs on an immune cell, you'll actually inhibit that immune cell. So we wondered, okay, could GD2 be binding to a Siglec since it contains sialic acid? So what we did here to identify the ligand for GD2 was we took a cell line that's GD2 negative. But that cell line is still coated with many different sialic acid containing molecules. So then you treat with some chemicals called sialidase, and it basically rips bare the membrane so that it doesn't express, so that there's no sialic acids left on the membrane. And it's represented here by a bare membrane. We then actually, you can just take GD2 that you can commercially buy, and because it has this long fatty acid hydrophobic tail, it'll actually just be taken up into the cell and sit on the cell as if the cell was, sit in the cell membrane as if the cell itself was expressing GD2. So this is basically a cell line that the only sialic acid it now expresses is GD2. And then you can buy soluble versions of all the human Siglecs. And we took them, and this was done by Benji Smith and Carolyn Bertozzi's lab. Without their lab's expertise in Siglecs and in glycobiology, we really wouldn't have been able to do these studies. And what Benji did was he took the soluble Siglecs and probed the cells that had the synthetic GD2 on their surface. And what you can see is of all these molecules, only one of them specifically bound to our synthetic GD2, and that was Siglec 7. And not only does it bind Siglec 7, but we can block that interaction by pre-treating with an anti-GD2 antibody. So what this tells us, you can see here, the binding of Siglec 7 to 2 neuroblastoma cell lines is really completely blocked by an antibody like dinutuximab, an anti-GD2 antibody. So Siglec 7 is expressed on macrophages and NK cells and is known to have some of those inhibitory domains. So it's an inhibitory molecule. And what this tells us is really for the first time that GD2 is also a don't eat me signal, that it's capable of suppressing macrophage phagocytosis. And it's through its interaction with the Siglec 7 receptor on macrophages. And so when patients are treated with an anti-GD2 antibody like dinutuximab, this really in itself is also enhancing phagocytosis by blocking this interaction. So when we think of our combination of anti-GD2 and anti-CD47, really what we're doing here is heavily tilting the balance away from anti-phagocytosis, or don't eat me, and towards phagocytosis. So first we use dinutuximab and an anti-CD47 to block don't eat me signals. And then also by giving dinutuximab, we're providing an Fc receptor to the macrophage, which drives it towards phagocytosis. And we're also upregulating calreticulin through that stress signaling at a cellular level. And this really drives phagocytosis by the macrophages of the cancer cells in our models. So I'll finish up just to show that this works in other diseases also. It's been known for a long time that GD2 is a target on osteosarcoma and a few other sarcomas, but in osteo, it's at much lower levels than neuroblastoma, but it is still pretty ubiquitous. There have been multiple trials that have tried to use antibodies like dinutuximab and others in osteosarcoma, and they haven't had kind of the profound effects that we've seen of dinutuximab in neuroblastoma. So in this model, what we did is we attempted to model really how humans end up with metastatic disease from osteosarcoma. So we inject the mouse with osteosarcoma in the hind leg, we let it grow for a month, and then we amputate the hind leg, and then we treat the mouse with antibodies, either GD2, 47, or the combination of both. And then a month later, we look at their lungs. And at this point, at amputation, the mouse has micrometastatic disease in their lungs. And this really is a model for the morbidity and mortality involved with osteosarcoma in humans. And so what you can see, and this is just looking at the lungs after removal, the tumor cells are GFP positive, so you can see them here. This is a group of untreated mice, or treated with a control antibody. And these are mice that are treated with anti-GD2 antibody. And what you can see is you still see these large tumors, not really a reduction in the tumor number or tumor size in most mice. CD47 results in much smaller tumors, but they're still present. And only the combination of GD2 and 47 results in a near absence of metastases. And so this is just the quantification of the metastases, but I'll say that, you know, there have been some trials, and there's some ongoing trials of anti-GD2 as consolidation for patients who have lung metastases removed for osteosarcoma. And seeing this preclinical data, it's not surprising that some of these studies have already presented that anti-GD2 is not effective in this setting. And we feel like we feel that you really need the anti-CD47 in order to unleash this compartment against osteosarcoma. So to conclude, you know, about anti-GD2 and anti-CD47, we think that this is a new way to go after pediatric tumors, really by unleashing the innate immune system, by tilting the balance of eat me and don't eat me signals. I think GD2 is now, we're working on newly defining it as an immune checkpoint molecule capable of suppressing macrophage activity. And this combination of anti-GD2 and anti-CD47, we're excited that with CTEP and the NCI, we'll bring this forward in a phase 1 multi-center trial in the upcoming year. So that's all the science I'm going to show today. And just before my acknowledgements, I want to show a picture of my friend Karen Glick, who died when we were teenagers. And I show this because Karen died from CML. She was diagnosed at the time with CML in blast crisis, and died in the year before the first studies of Gleevec were initiated. And I, like to show this picture because at the time that Karen was diagnosed, CML in blast crisis was an incurable disease. Now it's very much a curable disease because of Gleevec and other TKIs. And really what was previously thought of as really an incurable disease because of technology was able to become curable. And that's really what kind of defines the work our group here does, really trying to use new technology in order to drive the field forward, to take things that really seem incurable and create effective therapies for them. So with that, I want to acknowledge the many people who were involved in all of that work. My lab and Crystal's lab, Michelle Mangi's lab, Weissman, Bertozzi, Sorensen, and Weiss labs also. And of course, my funders, especially Alex's Lemonade Stand, who this year, actually funded the GD2 CD47 work with an A award. And I feel like they really put a vote of confidence into my new lab. So I just want to say thank you in this forum also. And then just a shameless plug that I'm always looking for motivated postdocs. So if you know anyone or you are someone that's interested, please just get in touch by emailing me.
Thank you so much, Robbie. That was a great talk. I really appreciate it. Congratulations on the A award. It's one of our hardest to get awards, and for many people it turns into being a career changer. So hopefully it does that for you.
Thank you.
I just want to remind everybody that we have another talk before we get into the questions. This is a week where we have 2 talks. So on Thursday of this week at 12 o'clock Eastern time. Usually they're at 1 o'clock, but this one's at 12 o'clock Eastern time. We have Mariella Philbin and Michelle Manji, who we heard a lot about on this talk, doing a talk about the seed in the soil targeting DIPG from multiple angles. So we hope that some of you will tune in for that. And with that, we're going to jump right into questions because we have a bunch that have come in. So the first one that came in asked, is there any concern that patients with brain tumors may have worse neurotoxicity from CAR T cells?
Yes, you know, I think when we think about the toxicity, potential toxicity of CAR T cells, you have all the things that have defined CAR T cell toxicity. So cytokine release syndrome, and then neurotoxicity, which in the case of leukemia and lymphoma is largely cytokine mediated. And then you have this new class when you start thinking about CAR T cells for you know, something like DIPG, which is in the pond of neuroanatomic toxicity. And I didn't have time to get into it in the talk, but in some of our models where we had tumors that were very deep in the tissues, we actually saw death of mice because of the inflammation that drove herniation. So you know, the clinical trial, we're kind of hedging against that with certain patients that will accept on the clinical trial, and then very close neurosurgical involvement. And this has been a real challenge and learning experience, and for something we're very fortunate to have our neurologist team and a neuro-oncologist and neurosurgeons. But it's definitely a possibility and a concern.
Okay, so the next question that came in is, are these studies done in NSG or immunocompetent mice?
So I think that, so all the CAR T cells are NSG mice. There are immunocompetent models of CAR T cells that are generally less used in the field because the receptor totally changes. So I'm going to guess, though, that the question was about the GD2 CD47, and everything I showed is in NSG mice. But with Bill Weiss's lab, I just didn't have time to get into it. We have shown in a immune competent model using a anti-murine CD47 that we get the same kind of the same effects, and that we really unleash the effects of anti-GD2 in that THMIC model by using the combination of GD2 and 47.
Then we have a follow-up from the same person that says, do you think limited anti-tumor activity of CD47 plus B7H3 antibody combination could be due to the mutated FC domain of MGA271?
You know, as far as I know, MGA271, I don't know the answer to that, but I do know that, you know, they've tested CD47 in Irv's lab and other labs at Stanford with many different tumor-specific antibodies, and some work better than others. And you know, my personal feeling is it's about the target, that, you know, it's the same reason I think that certain antibodies work in the clinic better than others. I think it's about the target, and the target needs to be, you know, be playing a biological role, as opposed to, it seems like B7H3, at least in pediatric tumors, is just kind of expressed, and, you know, blocking it with an antibody or just binding it to it with an antibody isn't getting the desired effect, like ligating GD2. But I think I should say that that's all kind of conjecture, and we haven't done the studies to know that. But I will say that in vitro, at least, we have an anti-GD2 where we mutated the FC to completely abrogate the effects of abrogate binding to macrophages, so abrogate binding to the FC receptor, and the effect is still there of the combination of GD2 and 47, really showing that these additional things like the upregulation of the calreticulin and the blocking of the Siglec are biologically important.
Okay. So next question is, are Siglecs expressed by other immune cell types? And if so, can they inhibit those cell types?
Yeah, that's a great question. Yes. That schema that I kind of ran through basically showed which types of immune cells they're expressed on. And yes, macrophage, NK, B cells, T cells all express Siglecs. And, you know, there's this emerging world of glycobiology, partially led by Carolyn Bertozzi's lab, that's shown that interfering in these Siglecs can also unleash immune cells. And I would point out that Siglec 7, besides being on macrophages, is also highly expressed, actually the highest immune cell on NK cells, which really are known to play a very important role in neuroblastoma.
Next question comes from Sid Mitra. Says, beautiful talk, Robbie. Very interesting work. Macrophage phagocytosis is also a precursor to immune suppression, especially in efferocytosis. Have you looked at immune suppressive cytokines after long-term treatment with GD2?
Thanks, Sid. I think, first of all, our models really just aren't set up to look at long-term treatment, but fortunately, the group that has run GD2 trials in COG has published work on cytokine production after anti-GD2, and I'm just not that familiar with the work and haven't taken a look, but I think we should.
Great. Next question, can mifemertide be used with anti-CD47?
I would assume anything could be used with it. We haven't looked at all at the level of synergy. Hopefully the Gilead and 47 Inc. and Gilead have gotten breakthrough designation for this agent in adult myelodysplastic syndrome and are going to try to bring it forward for a registrational trial. So hopeful that it gets FDA approved and then we can try all types of interesting combinations in pediatric trials.
Next question comes from Matthew Barth. I'm not sure if this is something you want to answer now or you want to answer through an email, but how would the multi-site trials for neuroblastoma or osteosarcoma be spread out and how can we get involved?
Sure. So those trials will be run through the Pediatric Cancer Immunotherapy Trials Network, which is a NCI-supported network of hospitals performing immunotherapy trials. The pediatric CITN is led by Crystal Makel. So these trials will be open through those centers. And then also, at least the 47 GD2 will likely open at NANT centers also, the NANT neuroblastoma centers.
Well, that brings us to that's our last question. So you made it through, Robbie. Again, we appreciate everybody joining us today. Another lecture on Thursday. Thank you so much, Robbie. You did a great job. We appreciate the lecture and appreciate all that you do to help kids with cancer.
Thank you. Thank you for the invite and thanks for everyone for listening.
All right, take care.